Good afternoon, everyone. Welcome to the Wednesday afternoon lecture series and a distinguished presentation today from Dr. Huda Akil, whose topic is The Depressed Brain, Sobering and Hopeful Lessons. Dr. Akil grew up in Damascus, and I'm able to learn, because she's been interviewed about this, that she was inspired by Marie Curie, who moved from Poland to Paris to follow her own dream of becoming a scientist. Hearing of those aspirations that Huda also shared of becoming a scientist, a family friend suggested that maybe she ought to dream big about going to America. Well, what do you know? That seems like what's happening, and what has been a great contribution to neuroscience as a result. So she got her undergraduate degree in psychology at the American University of Beirut, and also a master's degree there in psycholinguistics, and then did come to America to get a PhD in psychobiology and neuroscience at UCLA. After a postdoc at Stanford, where I gather she encountered some other people who are here, like Tom Insel, she then joined the faculty at the University of Michigan, where she has been continuously since 1978, rising through the ranks rather quickly, and now serving as co-director with Stan Watson of the Molecular and Behavioral Neuroscience Institute. I was fortunate to be at Michigan during 9 of those years, and overlapping with Huda was a very pleasant experience indeed, because of the open collaborative way in which she has approached all the science that she has undertaken. A major focus of her current research effort is both to develop animal models to understand genetic and developmental basis of differences in temperament, especially the implications for anxiety and depression, but also to study human postmortem brains and to try to identify pathways such as fibroblast growth factor that are involved in major depression, and I suspect she'll be talking something about that. She's a member of the IOM, the National Academy of Sciences. She's a winner of the IOM Sarnet Award with Stan Watson. She's been past president of the American College of Neuropsychopharmacology and the Society for Neuroscience. She's a NIH grantee over many years. I'm happy to say she's currently a member of the advisory committee to the director, so we will be keeping her very busy tomorrow and Friday with our current meeting of the ACD. Please join me in welcoming Dr. Huda Akil.
Excellent. Thank you, Francis. It's really an honor for me to be giving this lecture. I see many friendly faces, friends, even students, colleagues, past colleagues, collaborators, people who have agreed with us scientifically and people who have disagreed, and I love you all. So I also want to thank my ongoing continuing support for 40 years from the NIH, especially NIMH, NIDA, and then also the Office of Naval Research and the Pritzker Consortium. I think I received one of the very first postdoctoral fellowship that the National Institute on Drug Abuse ever gave. So maybe it was money not wasted. I hope it wasn't money wasted. So, and while I am thanking people, I definitely want to thank my collaborators, Stan Watson, who also is my husband, and we collaborate. Our students, trainees, we call them the WAKILs. It's a chimeric name for Watson and Akil. And I hope that you will keep an eye on their names in the corner of each of the slides, the scientific slides. I'm also going to talk about the results of work from this very interesting experiment in science, in a way. It's a team science called the Pritzker Neuropsychiatric Disorders Research Consortium. For a while, we also had a Conti Center, which was great, and they were very complementary kind of public-private collaboration. These are the universities involved: Michigan, UC Irvine, Stanford, Cornell, and the Hudson Alpha Institute, and here are the directors at each one of the sites. And it's been really fun because it's a mix of psychiatry, neuroscience, genetics, and there's about 100 actual scientists involved in this. And it's kind of it's a lesson in how to do things in a very different level, in a different domain. So as you know, my topic is about mood disorders, especially depression, and I don't need to tell you all this is a huge burden on humanity altogether and on this country in particular. And It's a very painful and hard to define illness. It's associated with severe psychological pain and a lot of dimensions of changes. Some of them are affective, but some of them are also cognitive, autonomic, motor, behavioral. There are lots of both physical, cognitive, and affective symptoms. There's multiple classes. Most of you know, of course, about both unipolar and bipolar depression. And one of the reasons the burden is huge is because this is an illness that happens early in life and can be lifelong, is not treated right. It's often underdiagnosed. It is not handled by the society, even by physicians. And it's stigmatized. And as a result, we really still have a long ways to go in finding treatment. A good chunk, 30% maybe of the people have treatment-resistant depression. As I was discussing with various colleagues today, it's not clear how much of this is genetic and how much of that comes about because of you run out of neuroplasticity and anything in between. And there is a lot to learn, and we have not had brand new types of antidepressants for a while. And a very big part of the challenge is that mood as a concept is not as easy to wrap your brain around, say, even compared to another brain disorder like a movement disorder. It's an ephemeral concept, and it's hard to even define it temporally. Like some people say, I'm in a good mood this moment, but when we talk about mood disorders, we're talking about protracted changes. I think of it just kind of operationally for me as a biological mechanism that we have that is adaptive way of telling us how we are doing in the world. Why should we have moods? What's the point of moods? And to me, it's a way of integrating information about where we are in the world, both internally, how our body is doing, and how we are doing in our environment. And if everything is humming along, then we're in a good mood. And if something is either happened that was bad or is threatening to be bad, then our mood is not so good. So moods then can also organize behavior, motivate you to avoid bad things, go to good things, and suddenly it starts linking with all these other functions that the brain has to do, cognitive, memories, affective, and the like. So when you have a mood disorder, then your prevailing emotional tone is distorted, and it is not necessarily well hooked to reality. It's lost this ability to give you accurate feedback about the world. So it's either stuck in a negative situation, and that is major depression, or it's oscillating in its own right and not coupled to the current reality of the situation. So you can think of it as an altered feedback function, and you can think of it as interfering with what I call emotional executive function. Your ability to prioritize, your ability to decide what should be in the forefront right now, what you should be doing using your brain as an executive versus what should fade because it's not so critical. So our efforts at understanding these illnesses have been rather difficult. I like the characterization that Mukherjee gave of the search for answers for cancer in this Emperor of All Maladies. He talked about the hypnotic drive for universal solutions, which we have definitely suffered from, and the queasy pivoting between defeatism and hope, which we continue to suffer from. So we really need to figure out how to wrap our brain around studying this complex illness, or probably sets of illnesses. If you ask Tom, he will tell you it's not one. And how to go about it in a realistic way, not trying to find easy universal solutions, but also not becoming defeatist about it. And as a basic scientist who's been in the business for over 40 years, you get to the stage where you ask yourself some existential questions. And to me, they're humbling. So I've wondered, have we really helped as basic scientists? As neuroscience, genetics, have we offered psychiatrists, clinicians enough information, enough substantial help in understanding all psychiatric disorders, but in this case, this one? And if so, why not? Are we deploying the right kind of animal models? Are we really modeling what we pretend to be modeling? Not just to get the grant money, but to actually understand and solve the problems. Do we have some blind spots, things that we don't think about, that we should be thinking about, even if they are hard. One of them I will give you is variability. I feel that basic scientists hate variability. They think of it as something to be ashamed of, whereas clinicians think of variability as something that is to be understood and embraced. So I will have done something about that by the end of the talk, I hope. And then I feel like we are too dichotomous. We want genes versus environment. And I think we need to come up, especially for these kinds of disorders, with a more integrated view. And we need to use all of this to try and understand more about causes and actual cures. But of course, the most humbling question of all is, are we smart enough? Am I smart enough? Very questionable, but it's worth a try. So one of the questions in whether we have been smart enough is, have we over-reduced the problem? So we've been, in neuroscience, we are always fighting the battle of stigma. And one of the ways that we have responded to the issue of stigma is that we have to tell people it's biology. It's not somebody's fault. They're not being lazy. They're not being just not motivated because they're trying to give you a hard time. This is a brain illness. And so that is definitely true. But then it's easy to say it's this chemical imbalance or that chemical imbalance. And sometimes we believe our own PR. And the other thing that I think we tend to forget is how dynamic this is, that people are living a life, that living a life with certain feelings changes behavior, behavior changes the brain, it changes feelings, and there is a cycle that develops over a life course. And as a basic scientist, I don't feel like we have taken care to model this well enough in our own animal models. So this is my usual kind of way of saying we can't jump from genes to behavior. We can't jump from gene to clinical. If there is a brain in the middle of your business, that brain is really in the middle of your business. It gets, it has a life of its own. So of course it is impacted by genes. It is changed by development. It's modified by hormones. In the environment hits it, for example, in the case of depression, oftentimes you need multiple stressors before you see episodes, but then the illness can take on a life of its own, and eventually some episodes start to happen, even if you cannot identify an external stressor. But the very important kicker is that the behavior goes back and remodels the brain, and these remodeling issues are now a few steps away from the original genetics, and all of this is kind of coming together in issues of diagnosis and prediction and treatment. So this is really why in this tension between should we study genes, should we study the brain, a few years ago, a few of us tried to kind of get together and write this article about the fact that maybe the area where we should all be focusing is more on neural circuit as an intermediary in the brain between where the genes and cells are integrating their activity and then what's driving the behavior. That it's important to not do an either or, but to find ways of integration. And I cannot tell you how happy I am that the Brain Initiative focuses on neural circuit. I think that group just got it absolutely right. Thank you very much, Francis, and the people involved. So it is very clear that different combinations of genetic variants can cause disruptions at the neural circuit level that can look the same behaviorally and clinically. It's also fairly clear by now that you can have the same set of genetic variabilities as far as we can see, but still get somewhat different outcome, and that we cannot get away from needing to look at it at a different level of integration. So we have been involved in the Pritzker Consortium in a number of studies, and what you'll see is that for me in this talk, it's not an either or, that we need to think about genes, we need to think about environment and animal models, and really my hope is to convey some of that kind of thinking style. So this is, you can see me? Can you see me here? I'm very important. So this is one of those papers with the psychiatric, genomics consortium that has studied 60,000 psychiatric subjects, and it's really been kind of interesting to watch what comes out of it. But what I really loved in this latest analysis, which I think has been very thoughtful and very interesting is that there are pathways that are emerging that are shared between different psychiatric disorders. And one of them is histone methylation. The strongest one is histone modifications. So these are epigenetic modifications. And the reason I'm picking them is to say that there is a genetics of epigenetics, which tells you that there is a genetic of responsiveness to the environment, which might be really at the heart of the matter of what we're dealing with in psychiatry. So here is an example of some pathways, entire pathways, as opposed to single genes. I don't expect you to be able to read them, that are characteristic of either bipolar, schizophrenia, or major depression. And what you see is there are some pathways that are actually unique to the classical diagnoses, like postsynaptic densities pops up as very highly affected in schizophrenia, but not in the others. But there are pathways that are also shared. Those red arrows are all pointing to histone methylation as a pathway that's altered in all three disorders. And even when you do combined analyses of individual genes and you do networks based on development, I don't have time to go into it, and expression, there are some interesting genes that pop up that I want to point out to you because they're going to come back. We came to those genes through our animal models and our human studies. This paper is 2015, so obviously it didn't drive the research, but I love the fact that we have some convergence. So these are calcium genes that pop up as being critical among those shared genes between the psychiatric disorders, more important in some than others. You can see FGF receptor, R1, is another one that I just highlighted. And you can also see HDAC4. So I just kind of keep them in the back of your mind. I'll come back to them later. So how do we go about taking something on like this? And very broadly speaking, in our group, we have looked at two approaches. One is more hypothesis-driven. What is it that we know about biology, stress, gene, environment, interaction, the creature in their environment? Can we come up with specific hypotheses and model them? And the other is more discovery-driven. And in all cases, our interest is to kind of go between animals and human, both post-mortem and living human, and we can start either in the animal or in the human, but the hope is to always go into the living individual. So I'm going to take just a few minutes to tell you about an example of a hypothesis-driven model, and I like it to just give you a couple of messages out of it. This is an inducible animal where we overexpress the stress receptor, the glucocorticoid receptor, only in the forebrain, so not anywhere in the periphery, in the hippocampus, in the prefrontal cortex, and it is inducible, so we can turn it on and off at will. And so you can see that the induction works, and it's very selective. This is the hippocampus, not eyebrows and eyes. And you can see it's pretty selective when we do turn it on, the transgene. And we did this study where we turned it on at different times. So this is the lifespan of the animal and their ages. And you can turn it on either throughout the life of the animal. We had a constitutive model, so we knew what to expect. So we replicated it by turning on the overexpression in this inducible one throughout the life, or only before weaning, or starting at weaning throughout the life. And all the testing is as adults. So these animals and these animals both have overexpressing glucocorticoid receptor. These guys haven't seen it for a few weeks. So what we see is with the lifelong overexpression, we replicate something we've seen before, that if you overexpress the stress receptor in the hippocampus, you make animals more anxious, which is not that shocking in that, this is something that denotes stress, and you should be nervous if you have a more active molecules that denote stress. So you can see less entries in the open arms, elevated plasmase, more latency to enter, multiple measures show it. Actually, animals are also more prone to depression, but they're very emotionally labile, which I have not shown you. They are also very reactive to antidepressants, so it's in that way they are more reminiscent of bipolar patient, anxious but labile and reactive. This is what happens if you express the receptor only before weaning and then shut it off, and it's completely gone when you are doing the adult testing. It is just as strong a phenotype as the lifelong expression. And this is what happens when you do it after weaning. They look much more like the wild-type animals. So there is a window in which you overexpress this receptor, and you get a lifelong phenotype. And after that, the system is set, and the animal seems protected. And when we do gene expression profiling on these animals, and we've done it in a couple of ways, one of the places where we see the biggest changes is in the hippocampus, in the dentate gyrus, as opposed to this other area, the nucleus accumbens. And we see some genes that are upregulated, some that are downregulated, and a lot of them are shared between the lifelong and the early life model. So telling us that these might be actually really important in the phenotype. The unshared ones may not replicate the phenotype. And here is what's interesting, is a lot of the genes that come up also have come up in some of the bipolar genetic studies. And I'm showing you an example of, this is one gene I highlighted for you in the network that I showed you before. It's one of those genes that is significantly altered in the lifelong animal and in the early life animal, but not different between early life and lifelong. And so it is a gene that might be important. So, and we have done some intervention in these animals to show what's functionally important or not. But the point is that we can start from specific genes, whether we think about them or we bring them from the genetic studies. We can create very brain-specific models, and we can start to study the function. And I've shown you an example: greater anxiety, depression, responsiveness to drug, and very early life induction, which I really was not expecting it to be nearly that clear-cut as it turned out to be. So for the rest of the talk, I'm going to switch over and talk to you about a more discovery approach where we didn't have a hypothesis. We went in a very open-ended way in the context of the Pritzker Consortium. So there we are involved in some of the genetic and genomic studies that are part of these big consortia. But we also have access to a brain bank, a very good brain bank at UC Irvine, where we do some of the analysis with different types of profiling studies. And when we started with this approach, we didn't pretend we knew where depression or schizophrenia or bipolar were. We have these three types of psychiatric disorders. We just took all brain regions we could get our hands on, and then we did much better dissection. So the Watson lab is really good at taking human brain and marking it with various markers, like this is with neuropeptides in the hypothalamus, and dissecting specific group with laser capture and doing some of the analysis. Sometimes the dissections are broader than that, and more we're trying to adapt the use of the clarity method to try and get a lot more bang out of our buck and see 3D if there are also morphological changes. This is working well in animals. It's still in the works for the human brain. So very early on, one of the things we saw is that there does seem, even with very old microarray technology, there was a different signature of the illness on the brain. So if you took the blue people that were a major depression, you saw, and you just looked across multiple brain regions, you saw that the hippocampus and the amygdala were had more significant differences from control, whereas in bipolar, even though the bipolar patients were depressed at the time of death, all of them, the bigger signature was in the nucleus accumbens. We have replicated this in a variety of ways with better and better techniques, including recently, this is from Rick Meyer's lab doing RNA-seq, where we're looking in much greater detail. But this is just a general slide that shows that, first of all, you can do clustering, and you can cluster the anterior cingulate and the dorsolateral prefrontal cortex together separately, very nicely separated from the nucleus accumbens. So that gives you some kind of faith that it's not complete, garbage. It makes biological sense. And at the same time, we can begin to look at correlation across three disorders. So anterior cingulate in bipolar, in schizophrenia, in MDD, DLPFC, nucleus accumbens. I'm showing you three regions, and I'm saying, are there correlations? And you can see that, for example, the anterior cingulate in the schizophrenic versus the bipolar have a lot of shared transcripts that are altered. fewer are shared between these two disorders. There are some, but fewer are highly significantly shared in the dorsolateral prefrontal cortex. And then fewer less in the nucleus accumbens. So this is Kevin Bowling and the whole group from Rick Myers and the Pritzker Consortium. Depression, less sharing, less evidence of what, that we can see of big changes. I think it's a lot noisier. So let's kind of back up and ask whether in the older data, the newer data, are there things that we can see reliably? And even in the very old data that we started, it was very clear with these differential signatures that in depression, what we saw most altered were growth-related, cell survival, cell signaling, cell death, cell cycle. They were in every brain region we looked at, and some of them were very profound changes. And even though the hippocampus and amygdala were the most altered, pretty much every region, and it was really not restricted to any single neurotransmitter system. It was very wide-ranging. And in some places in the amygdala, you can see actual just differences in the amount of RNA that we can pick up. So this is not published, but these are controls, major depressive patients who died, but not of suicide, and suicide, and you can see just at a very gross level, differences in what we can capture from them, done at the same time in a blind way. So, so very broadly, what is really clear from a 60,000 foot kind of point of view is that the dysregulation is wide. Nothing is like tenfold different. Things are twofold different, but there's a lot of changes, and a lot of them are in genes that I will tell you about, for example, immune genes. But what is really remarkable is that I have tried with various animal models to replicate this level of dysregulation. We never see it in our animal models. So this is like an end-stage situation that we're seeing. And it may have to do with treatment resistant. Maybe these are really sick people. Obviously, if they've died of suicide, they probably were not doing all that well. But, what we see is antidepressants actually change the gene expression. They change it to closer to normal, but they never normalize them and these people remain depressed. So on the power positive side, we have identified some things that we were not expecting to see. And these include, I don't know what this doing this, but beeping. Do you know anybody? So we've identified families of genes, immune genes, circadian clock genes, and growth factors that seem to be very clearly different in the brains of MDD versus controls. I am not going to go into all of them. I'm just going to take a minute to talk about the circadian genes because they are very interesting to me. Something is beeping. If somebody could help me with that, that would be good because it's driving me a little crazy. Can you figure out what that is? Okay, thank you. Appreciate it. So this is a very kind of dumb experiment in a way, but we did it anyhow, and it was one of those dumb experiments that actually give you something in the end. So we literally just lined up people around the clock based on when they died, and that's again thanks to Biff Bunny, who keeps very good records for his brain bank. So when you see a number, it means somebody died 1 hour after sunrise, 8 hours after sunrise, 12 hours under sunrise, like that. And then you just simply go to your gene expression data and you look to see if there is any evidence of change as a function of when somebody died. And what you see is rather remarkable. For example, here is Untitled, which is a clock gene, and you can see that it has a significant circadian rhythm. Just simply, these are different people, died at different times, all controls, but you can see that there is a rhythm that you can fit, and you can fit it way better, 24 hours than 12 hours, and you can do a lot of controls. And all the clock genes actually come up with some kind of cyclicity, even under these unideal conditions, and even their relationships are right. Like when PER1 peaks relative to PER3, that time difference is exactly what the animal models predict. It's the first time that people have been seeing it in gene expression in the human brain. Moreover, there is about 1,000 genes in the brain that show a 24-hour rhythm distributed all over the brain. This is not just in the suprachiasmatic nucleus. We're talking about outside of the SCN. So, and what is also interesting is that you now can ask, is there something wrong in the rhythmicity in any of the disorders? And we're starting to look at it in bipolar and schizophrenia, but the MDD is very clear. For example, here is a paper by Bunny et al. In Molecular Psychiatry last year. So when something is significant, it means it has a significant rhythmicity. And you can see these are classical genes that are part of the molecular clock, and they're all highly significantly cycling in the controls. And the matched MDD, matched by all kinds of criteria, all have no rhythm except this rev or alpha that has some rhythm left. But everybody else has lost their rhythm. Based on the pattern of gene expression across different brain areas, we can actually time the time of death of a given individual. So we can say, based on the signature in control brain, they died at 10 in the morning, and we are within an hour of the actual recorded death. So there is a timestamp on the brain as it dies based on these circadian clocks. And that is completely lost in the MDD. It is like they are been traveling in a different time zone. So those kinds of studies really start to tell you that we are looking at big orchestrated changes in the brain at every level, in their levels of expression, in their rhythmicity, in multiple circuits that we need to think about. But can we kind of hone in a little bit more molecularly? And my example for the rest of the talk is going to focus on the FGF system, where it was one of the first discoveries we made where we saw that this family, which I knew absolutely nothing about, was the most dysregulated in the cortex of depressed people. It seemed selective. It was more dysregulated than in bipolar or in schizophrenia. Since then, we've seen some effects, but not nearly as profound in these other illnesses. It's a complex system. It has 22 ligands and a lot of partnering proteins. It has only a handful of receptors, but the receptors are very complicated. They have splice issues. They have also partners. So it's a complicated system, the tyrosine kinase system. And these 10 years ago, more than, these are simple findings. Again, nothing hugely profound, but for example, here is FGF1, one ligand of the family, significantly decreased in depression, R2, R3. And if you look at people who had a history of antidepressant treatment, and we can measure antidepressants in their brain, so we do that in the talks, you can see that they lie right in between the control and the untreated patients. And very early on, we noticed that there are a bunch of dysregulation. It's not just one factor that was dysregulated, but the whole family was dysregulated. So, but one of the things that caught our attention is because there was a little biology done about, in other models on FGF and some of its receptors. We decided to start with that because there were more tools. And we noticed, so just to remind you, both FGF2 and its receptor, FGF receptor 3, that's selective. It interacts with other receptors as well. But they are decreased in several areas of the brain. Since then, other people have replicated that, and we have done many more brain areas. And so we asked, does this mean anything? Is this actually functionally relevant in any way, or is this just sort of a marker of something? So we went back to our model, but in animals, and we tried to look at each one of the facets of this schema and said, could it be a vulnerability gene? Is it a developmental gene? Is it involved in environmental interaction? Is it involved in neural remodeling? And I'm going to quickly go through all of this. A lot of this has been published. And the answer is we can use social defeat, repeated social defeat, as an animal model of depression, animals that lose repeatedly, even if it's a brief loss, and even if there is no physical damage, can start acting differently. They have fewer social interactions. They don't like sucrose. They show, quote, anhedonia. And they show these depressive kind of features. And when we use that model and we look at FGF in their brain, this is looking at the hippocampus, what you see is a significant decrease in the expression of FGF2 and some of the FGF receptors in the hippocampus, mirroring what we see in the hippocampus of depressed people. Some of those decreases are quite dramatic, like the dentate gyrus, and others are milder, but it is there after a few days of social defeat. What was interesting is we then asked, could it be an endogenous antidepressant? So we did a dumb study where we took FGF and we gave it to animals. And we gave it peripherally and we also gave it centrally. We looked at multiple models of depression-like behavior. This is the Poor-Salt swim test, which is sort of a bread and butter kind of boring test, but works. And what you see is, in fact, FGF2 acts like an antidepressant. So more immobility means greater depressive-like behavior in this test. More swim means less depressive behavior. And animals that received FGF2 peripherally or centrally, acutely or chronically, showed less immobility, greater swim, suggesting antidepressant-like effect. And interestingly, if you compete it with a small peptide, even online, in that moment, you can increase depressive behavior, suggesting that really any moment, in the moment that the animal is behaving, FGF is playing a role, because if you compete with it, it changes the behavior. And so then we asked, can we actually knock down the endogenous material and see the difference in behavior as opposed to giving it? Can we knock it down? And we aimed our knockdown to specifically the dentate gyrus of the hippocampus. And we, so the green is the lentiviral vector, and you can see the red is the nuclei, and the blue is glia. And it's mostly in the nucleus, in the neurons that it goes into the neurons as opposed to the glia. When we do the knockdown, we get a significant but not complete knockdown in the expression of the endogenous FGF2 in that region, no other region, very specific. And when we do that, what we see some changes in depression, but we see mostly changes in anxiety behavior. And this might be because it is very localized, and this area is important in anxiety. So you see these animals are spending a lot more time now in the closed arms of an elevated plus maze and less time in the more, scary areas of the plus maze. So we suggest here that, in fact, it seems to be important in the sense of, in the adult animals, it's part of the stress responsiveness. I showed you social defeat. I showed you that it's acting and modulating behavior in the adult. But is it a genetic factor? Is it a developmental factor? So in order to address that, we turn to this animal model that we have developed based on this idea that, there are no genes for depression. There is no genes for any one of those illnesses the way there are probably genes that predispose to certain styles of interacting with the environment that we call personality or temperament in real life. And there's a lot of epidemiological and genetic studies that show that some of these facets are predictive of certain vulnerabilities in the human. And so one of the major kind of dimensions is so-called internalizing versus externalizing styles. So and when people fall apart, get an illness, they get an illness that are kind of biased by whether you have an internalizing temperament or an externalizing temperament. So an externalizing disorder is one that's associated with so-called behavioral under control, where people seem somewhat less inhibited. They're more confrontational, they have more conduct disorders, they may have attention problems, they are maybe more prone to antisocial behavior or aggressive behavior, substance abuse. On the other hand, people who are more prone to internalizing disorders are more prone to behavioral over-control, depression, anxiety, and the like. So we asked, can we model that in animals and begin to use that and understand? And I'm going to get back to FGF, but you have to understand the model. So we started with these very simple experiments where you take an animal. I don't know if you can see the red, but this is ethovision, just tracking the movement of the animal. And I'm sorry you can't see it, but this animal is all over the place investigating this little object. It is a new environment in a new field with a new object, and he's all over it. And this animal is just hugging the wall. It's kind of a little scaredy cat, sits in the corner most of the time, never gets into the middle. So it's a locomotion in a new environment. How much do they investigate something new? So we just took this as a dimension, and we started breeding these animals. And we've now been breeding them for over 45 generations. And they separate like nobody's business, very quickly, very clearly. So by now, we have 100% certainty what their phenotype is going to be based on their lineage. This is not inbreeding. We started with an outbred stock, and we don't breed between families. But it separates very clearly, and the normals are somewhere in between. And we have the lineages on these animals. And we have characterized them on a whole bunch of behaviors, even though we bred them on this one dimension. And it's remarkable that a lot of behaviors come along with this simple breeding design, suggesting that we're tapping into sort of environmental reactivity style, and that, so aggression, depression, fear conditioning, and so on, all differ. So, for example, here is the elevated plasmase, the animals we call high responders who love novelty, also spend a lot of time in the open arm of the elevated plasmase. The low responders don't. It's not just because of motor behavior. We control for that. Expose them to stress and look at their social interactions. The high responders are really resilient. You can't hardly budge them. They stay very gregarious, whereas these poor guys, they just plummet pretty quickly. And it's responsive to antidepressants. And the fear conditioning, they have very big differences both in the acquisition and the extinction of fear conditioning. Those low responders, like internalizing animals, hardly ever want to extinguish their fear. I'm going quickly just to kind of give you a sense. This is where the outbred animals sit. They typically sit just in between the two. We're just kind of grabbing the tail ends of a normal distribution. And the high responders love to take drugs. They love new things, and they take drugs with great joy. And the low responders, it takes either a long time or some stress, and then they will take drugs. More importantly, if we addict them, even to the same extent, making sure they take the same amount of drugs, and now we look at relapse using various types of tricks, like after 1 month of abstinence, we give them a cue associated with the drug, and we look at them, we see big differences in them. So these animals, the high responders, love, as soon as they see the cue, they really want to start poking and taking the drug. So they show differences in addiction and relapse between them. And we have been collaborating with David Goldman here at NIA. It's a kind of a joint project with NIA and NIDA. David Goldman and Jiefeng Zhu, I'm sure I killed his name, to do whole exome sequencing on these animals. And we have data, and we have multiple genes that are interesting. We also have RNA-seq on these animals, so we're just getting our brain wrapped around this. So it's very clear there's a genetic differences. We've eliminated that it's not simply due to maternal behavior. We know that there is genetics involved. So the question is, if you have differences in temperament, do you need different treatments? That's what I call my precision medicine program for rats. And what about FGF? So I'm taking you back to FGF. And the answer is FGF is different natively in these animals. So the low responders have less FGF2 than the high responders, and there's a correlation between how much FGF you have in certain parts of the hippocampus and how much risk you will take in a new place or on the elevated plasmase. Moreover, if you give FGF2 chronically, you rescue these animals. You don't touch the high responder animals. They are already fine. They're not scared. The ones that are scared, you give them chronic FGF, you rescue them, and they become less scared. And you also induce neurogenesis in the process. So our hypothesis then is that this is a key molecule in temperament, that you can dial in the level of internalizing and externalizing disorder based on many molecular players, but that this could be one of them. And one of the questions is it changeable or is it set in place forever? So I showed you we can rescue it in adults, but can we do anything developmentally? And what we did is we gave it once on the second day of life. We gave one shot of FGF2 versus vehicle to both our high and low responders, and then watch them at different stages of life during development and in adulthood. We looked at neurogenesis, and we can see that FGF2 accelerates neurogenesis in animals, even outbred animals. And, but the most interesting thing is this little finding that says that, so here are, in the solid, how much time they spend on the open arms of the elevated plasmid. So how much risk they're willing to take or how anxious they are. And you can see now this is a later generation. You can see how different the high and low responders are in the solid lines. These guys are spending a good chunk of time out on a limb, and these guys hardly ever. Now, if you've given them FGF, nothing happens to these high responders. They're already out there. But the low responders actually change. Now, we're testing them as adults, and they had one shot of FGF when they were born, but now they are somewhere in between. So you can rescue a big piece of the phenotype, and we've looked at their addiction and their drug-taking behavior, and it just simply shifts them. It makes them also want to take more cocaine. So it's not like I'm not necessarily recommending it for everybody. What I'm saying is it's like you are somewhere on that phenotype dial, and FGF2 moves you away from the more depressive towards the more risk-taking phenotype. And you can do it once during development and change the program. And we have a whole bunch of molecules that we've looked through profiling that are mediators of these changes, that we are studying, that are downstream from FGF, that we think are mediating. The other thing I want to say is that even though the high responders don't seem to profit from early life FGF, if you do stress them, now you protect them. So this is inhibited social interaction. It says 100%, but it's inhibited because they've been socially defeated. Everybody's been inhibited by social defeat. If they've had early life FGF, now both the low responders and the high responders are made more resilient against social defeat by having had FGF early in life. So although you don't see the impact of FGF on the high responders under basal conditions, you push them, you start seeing it. So it seems to really be protective and kind of to be a resilience pathway. And this is for Tom. One of the downstream molecules is oxytocin. So when we give them early life FGF and we look at them in adulthood and see what changes, one of the molecules that really is induced only in the low responders is oxytocin, that used to be low and now becomes induced in various parts of the brain. So it'll be interesting to know how the whole program plays off. And part of the story is epigenetics. So how do these changes occur? Well, we know that one of the things we're doing developmentally is that we are mediating major, long-lasting epigenetic changes in these animals. And I don't have time to get into the weeds about it, but I think that those seem to be important in the induction of resilience. One of the targets that we have looked at especially is this H3 histone. It's methylation. It's not H4, but it's H3 K9ME3 at that site. And we looked at it because we saw some evidence that basally, just globally, there were differences between our lines in the levels of this histone. And we wondered if there were partnering molecules that were important. We looked as examples with CHIP assays at two molecules that I've talked to you about today, FGF2 and the glucocorticoid receptor. Remember that it is natively higher in the high-risk taking animal, the HR animals. FGF2 is anti-anxiety, antidepressant. GR is lower in these high-risk taking animals because, remember, it kind of signals stress. And what we see is we see signatures, natural epigenetic signature on these that are differential between our lines in multiple brain regions, where, for example, for FGF2, the inhibitory histone, modified histone, is lower in the high responders, whereas the activating one is higher. And exactly the mirror image for the glucocorticoid receptor. So it goes along with the expression is sort of all you want to know, is that there are epigenetic marks that are native before we do anything to these animals that go along with the differential expression. But the important thing is that we can change them. So if we give FGF2 in early life, we can flip them so that the animals that were more repressed become less repressed. The LR animals now have less of the repressive histone, for example, in the nucleus accumbens and in the hippocampus. They become more like the risk-taking animals. And remember, their anxiety is also changing. And more importantly, when we do knockdown studies of FGF2, we increase anxiety in the high risk-taking animals, high responders, and we also change the epigenetic profile accordingly. What we need to do is interfere with that histone and show what happens behaviorally. But our current hypothesis is that FGF does a lot of things, induces a lot of things, but part of what it does is it induces, during development, epigenetic changes that are then long-lasting and change the temperamental reactivity of the animal towards the environment. So I want to just wrap up by telling you, and this is sort of a model, that there is more than FGF2. We have a whole story with FGF9, which seems to be a functional antagonist, does exactly the opposite of FGF2. We're about to publish this work. So there are more than one target. And there are other FGFs in the story that are very interesting and partnering molecules. So there are multiple targets in this family to study. But the point is that here we started from the depressed human brains. We uncovered the new molecular antecedents, and we tried to take it to become kind of, to ask about its functional role. And what I really like is that a lot of this work has been replicated by others, including your colleague, Dr. Pinglo, and her lab here in a beautiful study showing the role of FGF in mediating some of the effects of Rosie here, and a mediator of the neurotrophin factor alpha-1. She showed exactly the same kinds of things that we showed about FGF being an important antidepressant and an important mediator of, so antidepressant and mediator of neurogenesis. I know I'm not, doing any justice to her work, but I just wanted to signal it. If you want somebody local to talk to, she knows a lot about it, more than I do. So but it's very good to know that people who do human work and different animal models are continuing some of this effort and showing that it is applicable in multiple ways in multiple places. I sort of came away from all this work with the idea that there might be these switch genes that are not the classical neurotransmitters, but that they are simultaneously a lot of things. They can be genetic factors, they can be mediators of environmental responsiveness, they can be growth factors, but they can be that sort of interface between the animal and the world outside, and sort of write these broader program within which then other regulation or dysregulation happens. And we're very interested, just in the interest of time, I'm going to skip some human studies to tell you that we're moving towards starting to use them as biomarkers in various stress conditions. So the lessons I have taken is that mood disorders lead to very persistent and widespread patterns of dysregulation throughout the brains that we're not really modeling very well, so we don't really understand very well. But there is a unique signature that could be secondary, not the original genetic signature. But the point is that we need to think beyond one transmitter. I mean, I'm not picking on serotonin. If you love serotonin, I love serotonin. But I'm saying we need to think about the whole ensemble of dysregulation that's going on, and then pick apart which symptoms, which dimensions, which temperament, which aspect we need to begin to analyze in a separate way. And that the same molecule is not an either or. It could be a genetic and an environmental mediator. But the good news is that your genes aren't your destiny. I've shown you an example where something is very genetic, but we could change it with some very early life manipulations. And I feel that these molecular organizers might actually be very cool targets for further drug development. And I still feel that we need to understand more about the dynamics of the illness, the sequences of things, and how to tap into it. So in other words, some of these genes, I think, are worthy of going back now and studying them, but thinking about them in a more dynamic way and with better models than we had in the past. And, my whole thing is that we need to face the music about the complexity of the brain and embrace it instead of, finding like it's just a headache and despairing from ever figuring it out. Thank you.
Terrific. Thank you, Huda. And we have time for some questions. The mics are in the aisles, so if you have a question, please go to the mic so that people watching by video can hear. Starting over here.
Hi. Sometimes viruses can use cell surface molecules like FGF receptor.
Yes, AIDS included.
Is anybody looking at infection and mood?
Yeah, people do, and we have a whole immune story, and the story about the immune system in the periphery and the brain are not necessarily the same, so we are just starting to look at side by side what some of the measures in the periphery and the brain show. But specifically, you're right that FGFs have been implicated in viral function and infection, and especially AIDS, and this is beyond my ken, but I would love somebody to work on it. Yeah.
So how you could translate the results in human patients? Obviously, you identified something. Is it translatable?
Is this translatable? Yeah. So obviously, as you could tell, it started in humans. So I'm pretty sure it's relevant to humans. So battle number one is won that way, by working backwards. So we and others have shown that there are some allelic variants that are also very predictive. My colleague Srijan Sen, for example, has genotyped people based on some various biomarkers, including some I've mentioned, could predict who's going to be depressed during their internship year. That I think is really a cool model. So as biomarkers, I think we're starting. What I skipped over was a Trier social stress test, a social stress test you give to humans, and we're trying to predict who's going to respond in what way at the endocrine level based on these biomarkers. So I think biomarkers is fairly proximal. We should be able to get at those if we just ask the questions right. In terms of drugs, we know it works if we give it to animals, but giving it to humans is a whole nother ballgame. One of my concerns is that FGF2 is very powerful, and it may be that it's too much to give. And one reason I showed you FGF9 is because it is working in the opposite direction, and it's better to get antagonists than agonists. So this is one direction we're going. And the other direction we're going is to look at partners of these molecules that are like more brain specific. So we would like to translate it, but kind of intelligently, hopefully, by trying to avoid some of the potential peripheral side effect, because these are very powerful agents throughout the body.
You are also looking at the human brain postmortem.
Yeah.
Do you see any effect of the FGF? Theoretically, it should be increasing the growth of certain neurons, mainly in the dentate gyrus and the hippocampal areas. So do you see any changes because of the FGF2 expression?
In the human postmortem, we haven't done that. We can't do that. One of the reasons that the Watson lab is focusing so much on clarity is to be able to take those brains that are very, precious. And if you take them and you grind them up for one kind of study, you can't get them. So we get one half to do one thing, the other half to keep for more morphological analyses. And it would be really important to get from the same brain as much morphological and anatomical data as we can get as many markers, including adult stem cells and other things, and certain other markers and so on in an anatomical circuit context, while we're also doing the genetics and the gene expression profiling. And if we could get all of that together, I think that would be good.
That's great.
Thank you.
You mentioned about the postmortem, and you did the brains, and you say you found different genomic profiles depending on the time of death.
Yes.
Okay. Well, there's been an awful lot of medical literature dedicated to circadian rhythms and disease.
Right. Yes.
And I can tell you from experience, a series which I published in the Archives of Pathology and Laboratory Medicine in 1998 on asthma deaths, is that one of the major findings was the asthma attack began in the early AM. So and that was the cause of death. Incidentally, in that study, we found one of the relations was between people who either abused drugs
Right.
or had at the time or had done it in the past, which is now becoming very big in the literature. A lot of people have caught on about it. The point is that your circadian rhythm seems to be, even though you pointed it out as an incidental finding, might be a major thing to focus on in terms of treatment.
And I mean, my colleague Beth Bunny has been talking about circadian rhythms and their role in affective disorders for many years, which is really what drove us to look at this thing around the clock and look at potential targets for intervention. I am not sure that the clock per se is broken in these people, at the molecular level, but I think definitely the regulation and there is still room to learn a lot about the patterns of change across the brain and how that might be influencing. By the way, what is really interesting is that when we lined up the depressed people around the clock, they were not as distributed around the clock as the controls. And I don't know what it means when people die. There was suicide and non-suicide in there, so the timing of when people die actually kind of is interesting. So what we had to do is then go back to the controls and pick only ones that died at about the same time in order to make sure that's not biasing the data. So I think even in depression, my point is that people die at different times than kind of randomly. And I don't know what that has to do with their circadian rhythm, but maybe.
Okay, thank you very much.
Yeah.
Huda, you showed this lovely data about gene expression and modification of histone methylation. But of course, much of your data has been by necessity focused on taking a piece of brain tissue.
Right.
We're all kind of excited about the idea of being able to go to single cells. Is that something that you think could change this picture substantially? Because you're obviously putting a lot of different cell types together here. What's the future of that?
Right. So, absolutely. So we've been working by successive approximation. The first one was, a brain region, and then I showed some slides where we were trying to see if we can grab molecularly defined, like pro-opiomelanocortin neurons versus CRH neurons or whatever. But I think eventually being able to look more at, especially given the level of mosaicism in the human cortex, it's asking if that is in fact in itself a variable. Whether you get more noise, for example, with some of these illnesses or less, and trying to see if we can capture that. So actually one of my junior colleague that we hired is now part of the consortium and is very interested in cortex and mosaicism and using genetic tools to look at that. So hopefully it will yield something useful. Yeah.
Well, we can continue the conversation at the reception, which is in the library. Please come and join if you want coffee, cookies, and conversation with Dr. Akil. But please, let's thank her again for a wonderful presentation.